So, hello everyone, this is Núria Queralt-Rosinach. I work at the Biosemantics group in the Leiden University Medical Center in Leiden, the Netherlands. And I'm here to present this poster about our work on modeling quantitative traits for COVID case reports. So this work was done in the context of the biohackathon devoted for the to fight the COVID-19 disease, this pandemic that affects globally all of us. So in order to support clinicians in taking decisions in the clinical settings, we need to understand how they monitor the observational patients in order to make these decisions and what kind of data they monitor, what kind of data they track. For that, we analyze the case report forms that track this observational data, and we found that they track quantitative traits like vital signs like temperature or blood pressure, or laboratory results like hemoglobin levels. These are quantitative traits. Typically, they are measurements measurements of parameters. So they track a measurement, a number, and a unit. And also they track all this phenotypic information, symptoms, and phenotypes. But the problem here is the need to capture and integrate this kind of qualitative and quantitative traits in order to understand and to make decisions in the clinics, but not only in the clinics for patient management, but also for research. So there is an ontology and a standard that is the clinical measurement ontology, but to encode this knowledge, to encode this kind of measurements, but the problem is that there are no links to phenotype ontologies. So the goals here were to formally describe and link quantitative assay data to phenotype ontologies, and also in order to help researchers to share found and access patient data to model these quantitative traits in a semantic manner, in a machine-readable manner. So we wanted to integrate these qualitative and quantitative traits. So first of all, we linked this ontology, the clinical measurement ontology, phenotype ontologies. This was one of the things, and the other was the development of this data model for quantitative traits. So the workflow that we follow, and it's one of the results of this work, is first we analyze these case report forms that we found that are very fragmented. Then we collect the terms and we map these terms to existing ontologies in the OBO Foundry. Then also we made formal descriptions for these links and for these models, and then applied these developed resources. And finally, we store all these to make these data and models and code freely available. So to link quantitative traits to phenotype ontologies, first we did this mapping of these terms to the clinical measurement ontology. Next, we did this development of these axioms in OWL. First, we decomposed the classes, the semio classes, using the labels, using lexical mining, and we decomposed these classes to three ontologies, to Uberon, Chebi, and Kebi, and Pato. Uberon is an anatomical ontology. Pato is a phenotype and trait ontology for phenotype qualities, and Kebi is an ontology used to describe molecules, biomolecules. So once we decomposed these classes, these classes into these terms in these standards, then we develop axioms using axiom patterns. We did this doing computable logical definitions for these classes. The approach was to use a compositional description of these classes, and then produce these patterns based on the EQ descriptions. There is the EQ means entity quality model that is used to describe phenotypes in phenotypic ontologies. So we developed these axioms and integrate all these axioms with Kebi, Uberon, Pato axioms, and mammalian phenotype ontologies axioms. And with these, we can apply these axioms using a reasoner like ELK to make inference using these old axioms, and to make relationships, to make connections between measurements and the kind of phenotypes that they affect. So for example, in here, I'm showing that we could establish a relationship between blood immunoglobulin E level to phenotypes falling under the immune system phenotype. So then we develop a simple model for quantitative traits. It's based on 4 ontologies: the unit ontology, the information artifact ontology, the BioC ontology, and the ontology by biological investigations. And then we integrate this model into the PhenoPacket standard for sharing genomic and healthcare data. In summary, we integrate quantitative traits with phenotypes for clinical interpretation biomedical research. We made this model for quantitative traits machine readable. It's a community effort, and we stored quote and data in GitHub that is freely available. So thank you, and come to visit my poster.